Hello and welcome everybody to today's GYSS Symposium. My name is Patrick Tan. I'm the Executive Director of the Genome Institute of Singapore, and it gives me great pleasure today to introduce our plenary speaker, Professor Michael Young. Dr. Young is a professor of genetics at Rockefeller University. I understand that many of you may be tuning in from different odd hours, so his lecture will be very relevant to all of you. Because in 2017, Dr. Young received a Nobel Prize with Jeffrey Hall and Michael Robash on understanding circadian rhythms using fruit flies as a model organism. His speech today is entitled Genetic Control of Sleep and Circadian Rhythms, and I'd like to pass the floor over to Dr. Young to begin his plenary lecture. So Dr. Young, over to you, please.
Thanks. Okay, so I hope you can see my screen. As was just mentioned, I'm going to be discussing the genetics of sleep and circadian rhythms. The two are, of course, related. Circadian rhythms, as you'll see, are much broader than regulators of sleep, but this is certainly how we arrived at this biological problem. Let me start by showing you some very old data. These are data that were presented by Pat DeCourcy in 1960 at a Cold Spring Harbor symposium that was on biological clocks some 50 years ago. And this shows running wheel activity for a rodent that is caged in constant darkness, constant temperature, with food available all the time so that they can eat and sleep at any time they wish. And the record that's shown here is starts at the top on day 1 and proceeds day by day, with each line being a different day, down to the bottom, the 25th day. These areas here that are shown as darkened lines are times that the animal is on the running wheel. So you've got an event recorder that's producing deflections in a pin that indicate when the animal's getting on and off its running wheel. And for many, I think what is quite amazing about this record, again in constant darkness, constant temperature, is that after very few days, you can predict within a couple of minutes when the animal's going to get on that running wheel and when it's going to get off of the running wheel. That this is an endogenous function is supported by the fact that these aren't 24-hour rhythms. Again, there are no environmental cues that we're aware of, but there could be something, of course, that's being missed. But then that would be inconsistent with the fact that each day begins a little earlier and ends a little later with regard to the time the animal's on that running wheel. So that it's a shorter than full day rhythm. It's an about a day rhythm, which is where the term circadian or circadian comes from. So this surely is, all the way back in 1960, indicated an endogenous rhythmicity. And there have been many hints before this that such rhythms were present in the biological world and were likely endogenous. This is an experiment that was done in the 1970s by Bill Schwartz, who was at the University of Massachusetts, and he looked at serial sections of a rat brain at 2 times of day. Either one collection of rats was sacrificed during the day, middle of the day, another group of rats sacrificed in the middle of the night. And these were rats that had been given access to radio-labeled water, radio-labeled deoxyglucose in the water. And what you see is overall there are different intensities of the autoradiograph day versus night, but most compelling is this area of the hypothalamus called the SCN for suprachiasmatic nucleus. And this was an area that appeared to be very active during the day and much lower activity at night. And this focused a number of anatomists on this area of the brain, causing them to do lesions of that area of the brain to see what the consequences might be. And they found that lesioned animals lost their circadian rhythmicity, no longer gave the kind of running wheel activities that I showed you on the previous slide. In addition, they were able to show that you could rescue the rhythms of such an animal in which this region, the hypothalamus, was ablated, by transplanting fetal tissue of the same sort from a donor animal into the ventricle adjacent to that to the missing SCN, and this was sufficient to restore circadian rhythmicity, indicating that not only was this a region essential for the production of circadian locomotor rhythms, but the ability could be transplanted in that sort of a fashion. Now, we know in mice and in Drosophila, for example, these are data from Drosophila, but there are hundreds of genes that are cycling with the circadian rhythm. So as the animal is behaviorally showing this rhythmicity, accompanying that is a broad rhythmic expression of the genome. These also are old data. These are data that we collected back in 2001. And what this represents are collections of RNA from Drosophila heads that are taken every 4 hours over a 6-day period. And what is shown here is each row represents the full time course of that 6 days of the expression of a given gene, with high levels of expression being shown in red, and low levels of expression, relatively low levels of expression, in green. And so over that 6-day interval, what we see is that the chart is organized so that genes that turn on in the early morning are near the top. Those at noon are shown in this region, those at dusk in the middle of the chart, midnight, and dawn again. So what we see is a progression, and I hope what you can see are 6 waves of rhythmicity moving diagonally across this chart from top to bottom. And the fact that these rhythms in this large group of genes is showing a rollout of gene expression in these diagonal waves suggests that at every particular time of day, there's a unique combination of genes that are hitting their high and low points with respect to expression. So what's shown here is the patterns of 150 genes, but many more genes are now appreciated to be rhythmic in this fashion, not only in the head, but in other tissues as well. Now, one of the consequences, or one of the things we understand from that kind of a pattern, is it becomes very significant with regard to thinking about targets for pharmacy. For example, we know that at this point in mammals, over half of the genome is rhythmic in some tissue. If you look from tissue to tissue, you will eventually find that at least half the genes are rhythmic somewhere. And in a 2014 study from John Hoganish's group, it was learned that of the 100 best-selling drugs in the U.S., more than half, 56 of them, were hitting rhythmic gene targets. Now, more significant than that is that of those that were rhythmic, half had half-lives that half of those drugs had half-lives that were less than 6 hours. So when you put those two features together, we appreciate that a large body of the drugs that we have that are most widely used are hitting targets that are oscillating, but being treated by drugs that have very short half-lives. So I think this is an area that's going to receive more and more attention as we understand better that timing of a dose as well as quantity is going to be important in in treating in a fashion that reflects more and more personalized medicine. Now, this is the chart that I showed before, again from Drosophila, with 150 genes stacked in this chart showing rhythmicity. And again, you see the rhythmicity in the form of these red and green stripes going from top to bottom. To the right is the same kind of a chart, but an abbreviated chart in which we're looking at 6 days of activity. And each day of activity is being represented, represents the results if we look at a particular mutation. On the left is a mutation that destroys the gene timeless, a null mutation for timeless. The second is a null mutation for the gene per. And finally, on the right, a null mutation for the gene clock. And what you see is that in each of those cases, the rhythmicity of that rhythmic pattern that we saw before is missing. And an important point, there are a couple of important points to make. One is that this is a very rare feature, that is to find genes in which mutations can abolish all of this rhythmicity. Pulling most of these genes out, for example, and eliminating them does little to the rest of the pattern. The other thing I'll point out is that for these genes that eliminate pattern, they do so even when even in the presence of a strong environmental cycle. So we're looking at light-dark cycles and measuring this lack of rhythmicity, indicating that although surely all of this was an adaptation long ago to a cycling environment, since the adaptation, since the evolution of these circadian systems, biology has come to be dependent on the interposition of a biological clock in order to interpret the environment. So finding genes and mutations that behave in that fashion required exhaustive genetic screening. And an example of the kind of apparatus that we've used in the past to find genes of consequence of that sort is shown here. What each of these cuvettes represents is a little lucite tube in which a single fly can be placed with a bit of food and a cotton stopper, and that's placed in a channel. On one side of the channel is a light-emitting diode, and on the other side, a phototransistor. And breaking, if the fly moves in such a fashion as to break that beam, we get a signal that can be collected by a simple desktop computer that will render an activity chart that looks very much like the rodent chart that I showed you at the beginning of this talk. And in this fashion, since we can do hundreds of these tests simultaneously, we're able to screen tens of thousands of genes for their impact on circadian rhythmicity in a single year. And as a result of that kind of screening, we now have about a dozen genes that we know are absolutely essential for the operation of these clocks. I'm just going to go through the simplest form of this mechanism that we understand at present. It's a cell autonomous mechanism. That is, it's a biochemical clock that exists in most cells, and it's formed by the action of the players that are shown here. Period is a gene on the X chromosome. Timeless is a gene on the second chromosome. And those two genes are coordinately turned on by the action of two positively acting transcription factors, clock and cycle, C and B in this cartoon. When these genes are switched on, they're of course sending messages out to the cytoplasm to produce proteins, a period protein on the one hand, a timeless protein on the other. But very soon after the synthesis of those proteins, we find there are two breaks in further action of this clock. One of the breaks is a casein kinase 1, which we call double time in Drosophila. This kinase physically associates with PER and phosphorylates it. And if PER is a lone protein, that is, if it's not bound to a partner protein timeless, then that phosphorylation leads to period degradation, a loss in the cytoplasm shortly after it's been translated. Another break, though, is caused by a protein called cryptochrome, which is a relative of photolyase, bacterial photolyases. It's a light-absorbing protein that, when activated by sunlight, will physically associate with timeless, causing both proteins to become phosphorylated, ubiquitinated, and degraded. So during the day, there's no timeless, and without timeless, period cannot be protected from double time degradation, and so we will not make this per-TIM complex. The per-TIM complex is crucial because it's only by making that complex that either of these proteins can move stably into the nucleus. Once in the nucleus, they can interact with cycle and clock and inhibit their activity, putting an end to that cyclical processes. And of course, when those proteins in the nucleus are degraded, this cycle can start once again. So I think you can see that in a day-night cycle, there's a basis for switching this mechanism back and forth between a nuclear and a non-nuclear regulation. But the delays that some of which I've spoken about also put an intrinsic oscillation into this feedback system that I won't have time to talk more about today. Now, I want to turn from Drosophila data to data on humans that we've been working on more recently, and specifically looking at certain sleep disorders that we thought might have their roots in alterations of components of these biological clocks in mammalian systems. And what I'm going to talk about today is a disorder called delayed sleep phase disorder. These are individuals that are night owls. This cartoon just gives an example of someone tapping away on a computer in the middle of the night. And of course, some of you may be listening to this talk in Europe, perhaps, at a desktop computer feeling just this way. But these are individuals that have a lifelong tendency to be night owls, to show this kind of phase-altered behavior. It's among the most commonly diagnosed sleep disorder in the US, possibly as high as 5%. Persistent delay in the timing of the major sleep episode, and resistance, there's strong resistance to efforts to advance sleep phase in these individuals. What's shown here are actigraphy of two individuals that were performed by our colleagues. A is a control individual, and B is an individual that had previously been diagnosed with DSPD. And there are two things going on here. One is that each individual is wearing an activity watch. They wear it all the time. And so deflections from the horizontal lines in these activity charts indicate when motion is being detected by that activity watch. And the individuals were also given a logbook and asked to enter in their logbook times each day when they get into bed and when they get out of bed. Now, the way the activity is arranged, or these plots are arranged, they're double plotted so that the episodes of activity are separated by the time when we expect the individuals to be sleeping. So this would be nighttime in both records. And what we see here for the control individual is that they're logging with these red triangles time that they're getting in bed, generally 11:30 at night, very consistently, and times when they're getting out of bed in the morning, generally 7, maybe 8 o'clock in the morning, consistently for that control individual. Now, if you look below it at B, the DSPD individual, of course, has activity that seems to range across the day and night. But if we look at the logbook, we see that this is an individual who is typically getting to bed quite late, always well after midnight, sometimes near or even after dawn. and getting out of bed at times close to noon. So this is an individual that is severely delayed and thus has this classification as DSPD. Now, we were able to study that individual and several individuals in that proband's family and a 3-generation kindred that's shown here. The proband is shown here. And what was asked is, what are the sleep behaviors of all the individuals in this kindred? And shown in the darkened circles and squares are individuals that show some level of sleep disorder. In black is shown a delayed sleep phase disorder. In gray, a related disorder that I won't go into until later. And over here in white is a cousin that has no evidence of a sleep disorder. So we performed whole exome sequencing on all the members that are shown here of this kindred, and learned that there was a single change that all the individuals affected by a sleep disorder shared that were not shared with the cousin that had no evident sleep disorder. And that was a mutation in Cryptochrome 1, human Cryptochrome 1, which we discussed before. And it's a single nucleotide change that has occurred downstream of exon 11 in its donor splice site. So with this substitution at this donor splice site, it's predicted that exon 12 will now be spliced to exon 10 with the loss of exon 11, loss of a 72 base pair or 72 bases in the ensuing RNA. These are RNA profiles, PCRs derived from amplifying RNA sequences from a number of individuals. These are from dermal fibroblasts from a number of individuals. This individual, number 11, is our proband. And this is the only individual from that family that is shown in this plot. And what you see there are 2 forms of the product being made that differ by 72 base pairs. Similarly, if we look at the protein made in those dermal fibroblasts, we've seen that individual number 11 is unique in producing 2 forms of the protein. So in order to determine whether or not, in fact, that mutation is causal, causes a change in the biological clock in those cells, we transfected naive mouse embryonic fibroblasts that were deficient in both Cryptochrome 1 and Cryptochrome 2, the only forms of Cryptochrome that mice have, with either the full-length form of the gene or this newly truncated form of the gene that we've identified. And what you see here in multiple experiments is that always we have an increase in period length, a longer period by about a half an hour is derived in cultured cells from the association, from transfection of that truncated form of the gene and protein. Now, I need to tell you a little bit more about how Cryptochrome works in mammals because it's a bit different from how it works in Drosophila. In mammals, Cryptochrome takes the place of timeless. Instead of being a regulator of timeless, it behaves like timeless in forming a complex with period, which is active in moving to the nucleus and associating with clock and BMAL to inhibit clock and BMAL activity on period and cry this time. So the other elements of this look very much the same as in Drosophila, but we now have an active role as a transcriptional inhibitor for cry in complex with PER. And for that reason, we wanted to know if there were changes in the way cry interacts with PER physically, or whether that complex of PER and cry has altered physical interactions with clock and BMAL. To do that, we transfected HEK293 cells with the genes encoding the protein shown here, a hemagglutinin tagged version of clock, a flag tagged version of BMAL, and a MIC tagged version of Cryptochrome 1 in two flavors. One, the full length form, two, the truncated form, or three, 50-50 mix of the full length and truncated form. So these are the inputs that we started with in this immunoprecipitation experiment. And now if we immunoprecipitate the hemagglutinin containing clock in this experiment, we find that we do pull down the full length form of Cryptochrome, but much less effectively than we pull down the truncated form, as is especially evidenced by the loss of the full length form in this from these cells that contain both forms in at equal levels at the outset. If we instead look at the effects on interaction with PER, we find that the input when we use PER as a target for immunoprecipitation looks very much like the pull down that we get when we pull down HA-PER, hemagglutinin labeled PER. So the interaction between PER and Cryptochrome seems fine, but there's a quite unexpected interaction between clock and BMAL and Cryptochrome. Now, we think we understand what's going on now from work that Carrie Parch at UC Santa Cruz has recently performed. She finds that exon 11 is a competitive inhibitor of interactions between Cryptochrome and clock. So there's a docking site on Cryptochrome for clock, shown here in green, and biochemical studies in which free exon 11 is allowed to associate with Cryptochrome will inhibit the ability of Cryptochrome to interact physically with clock. So it seems that Cryptochrome has a disordered C-terminal tail that includes exon 11, which appears most likely to interact in a dynamic fashion with that clock docking site to produce some level of autoregulation, auto-inhibition of that interaction with clock. And I'll say more about that later. So the next discovery that we made is that this is not a particularly rare mutation within populations worldwide. In fact, if we look at the exact database at Cambridge, in which we now have about 140,000 entries, alleles to look at. We find that in looking at these different populations, there's quite a variety in the frequency of this allele in those populations, but overall, just a little less than 0.5% of the alleles of Cryptochrome 1 that have been found in those populations are of this type, this Cry1 delta, as we often call it. And for example, since the carrier frequency, since this is a dominant mutation, the carrier frequency is what's going to be important. It would be twice the allele frequency. So for example, in Europeans, non-Finnish Europeans, we expect about 1 in 75 of those individuals will be carriers of this allele. So this gave rise to the opportunity to ask if we could identify additional individuals that carried this mutation, to ask about their sleep phenotypes. And in doing this, we collaborated with a laboratory at Bilkent University in Turkey, and were able to identify 6 families, large kindreds, that included carriers of this allele that could be studied for effects on sleep. So once again, what we're showing here are those that have delayed sleep disorders indicated by darkened circles and squares, and those that did not show a disorder indicated by open or white circles and squares. Most importantly, if we look here at the bottom right of this slide, if we look at the mid-sleep times for each of these individuals in this study, We find that the mid-sleep times are on the average around 4 in the morning, 4:30 in the morning for all of those individuals that showed no sleep defect, and about 6:30 in the 6 or 6:30 in the morning for the bulk of those that are carriers of this mutation, indicating a clear phase difference in the sleep patterns of these individuals. The carriers are more complicated. That is, there are individuals, there are outliers that have other forms of sleep disorders that we're studying now. So let me summarize what I've said at this point. So we have studies of several unrelated families where the presence of this novel allele, Cry1 delta 11, predicts delayed sleep phase disorder. And the penetrance and the frequency of the allele indicates that it could have a very broad contribution to DSPD worldwide. I'll also point out that since the allele is most prevalent in Europe, this probably is telling us something about the origins, the evolutionary origins of this particular allele. Since in the kindreds that we've studied, the mutation is found in multiple members of each family, all tissues should be affected, raising questions about what besides the sleep behavior of these individuals might also be affected by this altered running of the clock. Cry1 delta 11 shows enhanced binding to clock and BMAL1 in mice and in human cultured cells. And it appears to be a strengthened transcriptional inhibitor, again, due to the fact that it has an increased affinity for its transcription factor targets. So one of the interesting things to point out about this study is that study of a human mutation, because of the phenotypes it was producing, is also telling us something very basic about the function, how cryptochrome functions. It's elucidated for us functions that are associated with the disordered C-terminal tail of that protein. Competitive binding to clock and BMAL suggests a basis for its inheritance as a dominant trait. And finally, as I've shown you, Cryptochrome 1 delta 11 expression is sufficient to lengthen the period of mouse fibroblast rhythms. What I haven't shown you are human fibroblast rhythms, but they too are similarly lengthened when we use those as the target for those transfections. So let me close just by saying a little bit about where we're going with this. We think that these sequence databases are large enough now that they probably encompass most significant heritable variation, that is, most heritable variation that's associated with clinically significant disorders of sleep. And because it's so easy to count alleles and identify population-specific carrier frequencies, we think it may be useful to focus on candidate polymorphisms that are both prevalent and predicted to have an impact on the clock because of their association with protein modifications, for example, that we should be able to address large numbers of subjects and confidently determine human sleep circadian phenotypes, not only at cryptochrome, but at other known components of circadian clock. And of course, molecular mechanisms can be explored in cultured cells in much the same way that I've described for this variant of Cryptochrome 1. Finally, we know that prior hypotheses have hooked sleep and circadian rhythm disturbances to the etiology of a number of clinically important human disorders far beyond sleep. And a higher allele frequency, we think, brings unique opportunities to assess the impact of mutations of this sort on additional phenotypes. Again, all the tissues should be affected in these families by these mutations. So the families become great resources for asking what other effects on physiology, what comorbidities these mutations might bring with them. So I'll just close by indicating those that have done the work I've been speaking about. Alina Patke is a research associate in the lab who's been working on the project the longest and has done by far the most work. All the molecular biology that I described was done by Alina. Scott Campbell and Patricia Murphy originally identified the proband and that family, initial family that we studied with DSPD. Anna Krieger, also at Weill Cornell, helped us evaluate the sleep and activity records that I described for our DSPD and control subjects. Tayfun Osalek and his student Amir Anat helped with the Turkish studies. They identified the families that we studied and helped with the phenotyping that we presented. So with that, thanks very much. Let me see if I can stop.
Thank you. Thank you very much, Dr. Young, for that fascinating lecture. There are a number of questions that have come in, and they are broadly defined into 3 categories, and we have about 10 minutes to go through them. One category related to the impact of external processes on the circadian rhythm, one on the impact of these on human pathology, and finally, a more specific question about the mechanisms. So I'll start off with the first broad series of questions. Dr. Young, can you comment on the ability of changes in the environment to impact the circadian process? So for instance, coffee, temperature, the influence of COVID-19 lockdown, and so on. What sort of impact do these cues have on this natural cell autonomous process?
Right. So let me start with something like jet lag, which is familiar to many of us, maybe all of us. This is a demonstration of how environment can have a very profound effect on resetting these clocks. But more importantly, I think if we think about what is really being affected when we change a time zone, it's not just our behavior that's being affected. All of those genes that we spoke about are being altered with regard to their phase when we shift time zones, or with daylight savings time, for example, or the shift away from it. And each tissue has its own collection of rhythmic genes that are responding in that fashion. And what we find is that some tissues respond to different environmental signals during jet lag. So for example, your brain, the SCN, is quite responsive to the change in lighting that's associated with moving across time zones. Your liver is much more sensitive to shifts in the schedule of feeding, of access to food. And in fact, if there's a conflict between your liver and your brain, or if there's a conflict between the timing of food and the timing of light, your liver will listen to metabolites before it'll listen to your brain telling you what time of day it is from the light-dark schedule. So the environment has an impact on these clocks in many ways. There are biological molecules that we produce that also interact with these clocks in ways that can reset them, and interact and thereby interact with the impact of environmental factors. So there's a very complex mix of instructions that are coming from the environment and also from within that produce significant changes in the phases of these clocks in tissue by tissue fashion.
Thanks very much. We'll now go on to a separate series of questions about the associations between disruptions in the circadian cycle with impact on human disease. Can you comment a bit upon the association with, for instance, mental health, chronic disease, and are we getting enough sleep?
Right. So this is something that we're starting to look at now, and I in the talk suggested one way of doing this, which is to say if you want to link a physiological disorder or a behavioral disorder, a mood disorder, for example, a psychological disorder. If you want to know what the impact of altered patterns of sleep, for example, or altered patterns of circadian rhythmicity are, then if we have a series of circadian mutants, if we have variants represented in a human population that we can study in family form, in groups of families, where we have segregating within the family those that have an allele and those that don't, then we should be able to be pretty confident about whether or not another aspect of the individual's health is affected by that circadian clock and what that aspect of health is. But so far, everything is still under investigation as opposed to hard and fast proven. There are interesting discoveries that are being made, though. For example, our ability to heal, if we look at wounding, wounding is a circadian. It responds in a circadian fashion. So if you're wounded during the day, if you're a human, if you're wounded during the day, if you get a burn on an arm or a leg during the day, that tends to determine the rate at which recovery will occur. So there's a significant difference in the rate of wound healing if the wound occurred during the day versus the night. In mice, which are nocturnal, it's just the opposite. A nocturnal wound will heal significantly faster than a wound that occurred during the day. So there are many. There's a landscape of these kinds of findings that are emerging. Many of them are well known to us. Cardiac health, for example, its importance with regard to time of day. There are studies that indicate that cardiomyocytes are much more labile, much more subject to stresses at certain times of day than in others. Those are just a couple of examples, and there are many more, but none of them are really nailed down in a fashion that I would like to see, which is using the genetics and studying large families.
Thanks very much. We'll close off the session with a few questions related to specific research topics. So there's a fascinating question here about behavioral science on model systems. And the point here that a questioner asks is, we do the assays on mice and rats during the day when we are awake, but that's actually when they are asleep. And does that change the impact of these behavioral assays? Comments on that?
Sure. Yeah, I think that's a big problem. And I mentioned very briefly the fact that we have so many drugs with short half-lives that are so widely used and yet are hitting rhythmic targets. And I think that's a sort of an indication of some of the problems that you run into with regard to the kinds of problems that you were mentioning. So as we find out more and more about targets of these drugs and their oscillations, I'm sure we'll come to a point to understand much better what kinds of attacks we have available to deal with their management.
Thanks.
I think that's a key area that we'll see in the future.
I think we'll, in interest of time, we'll close off with the last question for today, which is, you mentioned in the very start about that many of these drug targets are actually following a circadian pattern. Are you aware of any pharmacological or technical approaches to reprogram expression in these target tissues so that we can reactivate their drug targets at the right time?
No, I'm there, you looking at the components of the clock, you could imagine that you could have a kinase inhibitor, for example, that was specific for casein kinase 1. and you should be able to have an impact on the clock. But the question is, what tissue do you want to hit? And how many other functions does casein kinase 1 have? So I think these are tough issues. The other point that in the last question that I failed to get to, which I think is very important, is the use of mice, for example, to look at to do pharmacology and other studies of interest to health. And it's certainly true that it's going to be important to think about comparing active phases of these animals. We could be missing the effectiveness of a drug quite completely if we apply the same time of day to a mouse experiment and a human pharmacological treatment. And so I think that's something that's going to need increased attention in the future.
That's an excellent point. And thank you very much for your time with us. And we will call this session to a close. Thank you very much.
Thank you.
Thank you.
Bye-bye.